Hey there, ladies and gentlemen. Welcome back for another lecture where you're now officially starting Unit 4. So we're on our home stretch here. We're going to start today with Lecture 17. We're going to talk a little bit about the central and peripheral nervous systems and start building some connections. So the whole idea with this unit is that we're going to take what we've previously learned in Unit 3 as well as 1 and 2 and really try to tie in a lot of a greater meaning. So if you remember back to Unit 1, we talked about being able to see the human body from an atomic level all the way up to an organismal level. And so this unit focuses on taking those connections and taking that information that we learned in Unit 3 about how neurons work, how action potentials travel down a neuron, and stepping back a little bit to see how do these messages move within our entire body and how does our body respond to these and process those. So again, we're kind of talking about sensory information coming in through an afferent pathway, being integrated, and then some kind of control being transmitted out to our bodies. So let's go ahead and get started. Our first handful of slides here is going to be just a real quick overview of our nervous system. And so if you remember, very beginning of this class, we talked about the endocrine system and the nervous system and how these two systems are maintaining coordination of our bodies. And so our endocrine system is doing that through chemical messengers, which we call hormones, and the nervous system is doing that through electrical and chemical means. So we've looked at that just recently. We looked at how the neurotransmitters are sent across our synapses and how that stimulates an excitatory or an inhibitory signal in the postsynaptic neurons. If it's excitatory, then we get an action potential that's transmitted down that axon. And so basically our nervous system then is going to receive and transmit information, process information, and then issue commands or controls to our effectors. Now, we talked about our different divisions of the nervous system. We said our central nervous system is our brain and spinal cord, and today we're going to look at some of the pathways through that central nervous system. And the peripheral nervous system, which is everything else that comes off of our spinal cord. And we're going to take a little bit closer look at where the peripheral nervous system and the central nervous system unite and connect. Now, here we had mentioned, this was a slide that just kind of illustrated the divisions of all of these systems. And so in our central nervous system, we have our brain and spinal cord, and in our peripheral nervous system, we have all these other divisions. So we're going to be looking at how these two unite and how that information is transmitted in between those two systems. The properties of neurons we had mentioned, they're excitable, so some kind of stimulation will excite them. So whether that's some kind of chemical signal, some kind of electrical change, or some kind of mechanical deformation, that's going to stimulate those neurons to fire their action potentials. They conduct, and so they'll transmit those action potentials down the axon and across that synaptic cleft. And at that synaptic cleft is where we see their secretory properties, where they're going to release different types of neurotransmitters to have some kind of an effect on that postsynaptic neuron. And they do that by binding to different types of receptors. Those receptors then open in response to those neurotransmitters, allowing different ions to move in or out of the cell. When we talked about homeostasis originally in this class, in Unit 1, we sort of introduced the ideas of an afferent and an efferent pathway. And as you can see here in this diagram, we've got our peripheral nervous system. So we have our sensory neurons, or our afferent neurons, that are going to take those signals from whatever parts of our body, and they're going to transmit that back into our central nervous system. We see that there's some kind of connection, a lot of times with interneurons, and then that signal is sent back out through our motor neurons, or our efferent pathway, to have some kind of effect either on our muscles or our glands. Here's that image of our neuron that we spent a lot of time going over and really learning about all the different structures that are involved with this. We got real close and looked at the ion channels that exist along the dendrites in the cell body and along our axon, and we looked really closely at those synaptic knobs to see what does it look like, what's going on at those synaptic knobs with our neurotransmitters. So again, spend some time refreshing over this, making sure that you're, got this dialed in. Then we also talked about the variability in neuron structure, and so we had different types of neurons. So we have our multipolar neurons, our bipolar neurons, which we'll see in lecture 18 when we talk about our sensory systems. And then our pseudo-unipolar neurons, and these are our sensory neurons that are coming in from our skin and our organs. And then our anaxonic neurons. And we won't spend much time discussing those, but these other three are sort of the more common ones that we'll be going over. And then if you remember our neuroglial cells, we have different types of neuroglial cells. We had 6 in all, but 4 were located in our central nervous system. And so here we can see those oligodendrocytes. They can branch out to multiple axons of multiple neurons. They wrap around those and they are producing the myelin sheath to insulate that action potential and speed up the transmission. Our ependymal cells are going to be producing that cerebral spinal fluid. Our microglia, which are kind of scanning the brain tissue, phagocytizing foreign debris, and kind of playing a role in our immune system. And then finally our astrocytes, and those astrocytes, as you can see here, have those paravascular feet that are surrounding our blood capillaries. They're also forming a framework or a supportive framework and communicating with those neurons as well. Now in your labs you'll be dissecting a brain, and so you'll be learning about our ventricles, and you'll see the pathway that cerebral spinal fluid takes along its journey. We'll see these 2 lateral ventricles. On this image we're just seeing the left lateral ventricle here. Over here on the right side we can see that right lateral ventricle. In the middle between those we have our 3rd ventricle, and deep to this image is our 4th ventricle. Along the way we have a structure called the choroid plexus, and the choroid plexus is lined with those ependymal cells, and this is where our cerebrospinal fluid is being produced. And on those ependymal cells we see little cilia that are helping to beat and wave and create a little bit of a current to move that cerebrospinal fluid through the brain and down and around our spinal cord. So in general, if we look at a capillary, we can see that the cells forming that capillary wall have little pores in between them, and that allows for substances to leak out. Our brain receives about 15% of our blood flow, which is about 750 mils per minute. And these neurons, remember, have a high demand for ATP, and so they're using about 75% or 70% of their ATP to regulate that resting membrane potential. So if they're producing lots of ATP or using a lot of ATP, they need to make a lot of ATP, which means they need a lot of oxygen and glucose. And so in doing so, by supplying the brain with oxygen and glucose, that means we can also deliver things that are toxic to the brain. And so these astrocytes here are surrounding those capillaries in our brain, producing the blood-brain barrier. And this helps to force these capillary cells to tighten and have tight junctions between them to close down those pores, so that anything that is in the blood has to pass through our blood capillary cells and those astrocytes, so we can reduce unwanted things into the brain or reduce toxins into the brain. So our blood has a lot of antibodies, a lot of macrophages, but it also has toxins and other harmful substances in there. So this blood-brain barrier system is going to regulate that, the entrance of those harmful toxins. So where can these things get in? Well, they can come in through those blood capillaries, or they could come in through the choroid plexus, which we just mentioned a little bit more, and get into that cerebrospinal fluid. So again, these astrocytes are inducing those cells, those endothelial cells, to form tight junctions, and so that's causing any substance to move through those cells and not between them. And this way we can reduce the likelihood of substances moving through. Now, that blood-brain barrier system is highly permeable to water and glucose, like we mentioned, but also lipid-soluble substances. So oxygen, carbon dioxide, alcohol, caffeine, nicotine, these things are able to move through. But it's an obstacle for antibiotics or for chemotherapy drugs. And so that causes an issue if that person or an individual has a brain tumor. Now, to again tie in a little bit of information that we talked about before, remember, tumors are the result of hyperplasia, and so our mature neurons don't really split, right? They have little or almost no capacity for mitosis, and so those cells are not what's forming the tumors. Where we see tumor development is in our meninges, or most of which come from our glial cells, but also metastasis from other areas in the body that migrate into the brain. So a glioma then is a tumor in those glial cells, and because of that blood-brain barrier, it makes it really hard to deliver treatment or deliver drugs to affect that tumor. And so a lot of times treatment then means we need to utilize radiation or some type of surgery. Again, a little blast from the past, some neural coding information. We talked about this on the very first day of this class. We alluded to it again in Unit 3, and so here it is. Again, if you remember, this is the way that our nervous system is processing information, or taking information and turning it into some kind of meaningful pattern in the brain, so that we understand how often action potentials are coming in, or the strength of those action potentials. So qualitative information is which neurons are firing, and quantitative information is that frequency or the intensity of that stimulus that's coming in. This helps our bodies and our brains understand what's happening. And we do so through different types of circuits. So we went over these earlier, but we have diverging circuits where one neuron is receiving information, but it branches out to a lot of different areas. The opposite then with converging is where we get lots of sensory input converging in on one neuron. We got our reverberating circuits, which we talked about with short-term and immediate memory, and then that parallel after discharge, where we have different information being transmitted in different times, so that we get sensory information that's consistently coming in. And the example we used was with our eyes, and that's why we don't see when we blink. And then we talked about synaptic plasticity, and we talked about how taking information and strengthening those pathways, reusing those pathways over and over and over again, kind of like driving your car to work or walking to work, or taking the same pathway over and over again. Eventually you get really good and you know what landmarks are around, and you don't really even have to think about it anymore. The same is true with our memories, and we can take that information and transmit it along that same pathway, and that's going to strengthen and increase that transmission over time. And so we call that synaptic potentiation. So this is where we are having this increased excitability and a long-term functionality. And when we talk about our long-term memory, hopefully what you've been doing all semester is striving for long-term potentiation with this information. And we're changing receptors, we're increasing the transmission speed, we're creating a longer lasting effect. Now, if we look a little bit closer at what long-term potentiation looks like at those synapses, it's pretty interesting how our bodies work and make this happen. So up here, we can see that we have our presynaptic cell, and if you remember, that action potential will travel down that axon to our synaptic knob, where that will open voltage-gated calcium channels. Calcium moves in and initiates the release of different types of neurotransmitters, and in this case we can see glutamate here, one of the most common excitatory neurotransmitters that we have in our brain. Now, what happens is that glutamate is released and it's going to bind to sodium and calcium channels, and you can also see that in this receptor we're allowing sodium and calcium to pass through. You don't need to know the specific name of these receptors, AMPA and NMDA. You don't have to memorize that. I'm not going to ask you about that. But what I would want you to know is that glutamate is binding to sodium and calcium receptors. That then allows sodium to pass through those channels, and in this receptor over here you can see that there's a darker, bigger arrow, and that is sort of designating that more calcium is going to come in than sodium comes in. And so here, calcium is coming into the cell, sodium's also coming into the cell. Both are positive, and so they're both going to create an EPSP. So if we look at calcium then, as calcium is coming in through this receptor, it is going to then initiate this CAMK2 pathway. But what we see is that is then inducing an increased amount of sodium channels to come through these sodium receptors. So those receptors are going to stay open more and allow more sodium in. At the same time, we see the production of nitric oxide, and nitric oxide is going to increase the release of glutamate from that presynaptic axon. That glutamate is going to stimulate the opening of these channels, more sodium and calcium. Calcium's going to initiate this process, allow more sodium back in, more nitric oxide to release more glutamate. And so what we see here is a strengthening of this connection between this neuron and this neuron. And so we're going to continue to get excitatory signals that are going to help really reinforce this pathway. Now, some of us probably have family members that have experienced Alzheimer's disease. The prevalence is about 100,000 deaths per year from Alzheimer's disease. We see it in about 11% of our population over 65 and 47% by 85. It's characterized by a memory loss, kind of moody, combative. They lose the ability to talk or walk or eat. Researchers think that Alzheimer's disease is a result of some genetics, but also deficiencies in acetylcholine as well as nerve growth factor. And as you can see in that image on the right, we can't really confirm it until we see an autopsy and we can see that severe degradation of those neurons in that brain tissue over on the right side here. So we have that atrophy of those folds in that cerebral cortex there. Some treatments that they've been trying is to give nerve growth factor or cholinesterase inhibitors. And if you remember from our last unit, acetylcholinesterase breaks down acetylcholine and shuts off that signal. So if we block that enzyme, it shouldn't break down acetylcholine, and therefore acetylcholine should remain in those synaptic clefts longer and still have an effect on those postsynaptic receptors. So go ahead and take a few minutes, practice these questions, think about what we just went over, and I'll see you back here. Welcome back. Hope that went well for you. Hope you were able to digest that a little bit here. So moving on, let's take a step back now and let's really actually start to dive into our connections here and look at the meat of this lecture. So we're going to start with looking at our spinal cord and our spinal nerves, and then eventually we'll talk about some reflexes. So our functions of our spinal cord, one is that it is involved in conduction. And we know that because we know that our spinal cord is made up of neurons, and we know that neurons conduct signals. So what we see here with our spinal cord is that this is sort of like the main freeway passing through our body, right? Information is going up, information is coming back down, and it's going to connect our body with our brain. We are going to see that this helps us with locomotion, right? And it's going to communicate signals with our muscles so that our muscles can move and respond to what we're trying to do. And we'll see that it's also involved in reflexes. And reflexes are involuntary and they're stereotyped, meaning that they happen pretty much the same way every time. Now, if we look at our spinal cord, where does it start? If you remember from your axial skeleton lab when you were looking at the brain and you had the foramen magnum at the base of our skull there, inside of our foramen magnum, we have our brain stem. Outside of the foramen magnum is where our start for our spinal cord exists. That's going to pass through the vertebral canal and all the way down to L1. Branching off of that spinal cord along the way are different spinal nerves. And so we have 31 pairs of spinal nerves that are going to come off on the right and the left side. And so we can name them accordingly to where they're coming out. So C1 to C8, and those are going to come out through those intervertebral foramen in between our vertebrae. And so we can name those along that segment. So we have T1 through T12 and L1 through L5 and S1 through S5. So just like we named our vertebrae, we can also name our spinal nerves coming off. And these are going to go to different regions of our body, different organs, different parts of our skin. Now, if we look a little bit closer at the meninges, and the meninges, remember, are going to surround our brain and spinal cord, and they're going to contain that cerebral spinal fluid. So here we're looking at a superior view of a superior cross-section through our cervical vertebrae and our spinal cord. And we know it's cervical because we have that bifid spinous process. And we have our transverse foramina over here and our transverse processes. And so we can see that our spinal nerves are coming off through those intervertebral foramina. Now, we can look at those meninges, and the three layers we have are the dura mater, and so this is the most superficial layer. We can see it surrounding the spinal cord here in the center, as well as part of that spinal nerve. We can see the arachnoid mater, which is deep to that, and directly in contact with our spinal cord is the pia mater, and that's that little pink layer right there. Now, in between the pia and the arachnoid mater is the subarachnoid space, and in that subarachnoid space is where we find our cerebrospinal fluid. Now, you'll also see that in this image, I've got highlighted this epidural space that has fat tissue in there, and that's going to help insulate and protect that spinal cord from bumping against the bone on this vertebra. Now, during fetal development, we can have various defects that occur, and one of those is with the vertebrae. If they fail to form, or the vertebral arch fails to form and enclose that spinal cord, we can get a bulge with those meninges and part of the spinal cord. Now, there's different types and varying degrees of severity with this spina bifida, but we generally see this happening in about 1 in 1,000 kids, and we see that it's common down here in that lumbosacral region. Generally, this is happening within the first 4 weeks of development. Now, one way to prevent this from happening is to have a really healthy diet, but also you can supplement with folic acid. And because this is only happening, because this tends to happen within the first 4 weeks of development, and a lot of times women don't know that they're pregnant for at least a couple weeks into development, is that you need to supplement your diet 3 months before conception. So this is where family planning is really important. And so if you're actively having intercourse with a partner and you're not using protection, or you're actively trying, you should be taking folic acid supplements just to ensure that you have a really high amount of B vitamin in your diet, so that will help prevent any spina bifida issues. All right, now a few other terms. Just to clarify, as we move through this section, you'll be seeing these different terms in bold over here to the right. In the central nervous system, a collection of cell bodies, or those somas, is called a nucleus. A bundle of axons in the central nervous system is then called a tract. While in the peripheral nervous system, we use the term ganglion to represent the collection of cell bodies, and a nerve to represent a bundle of axons. So when we see nerve and tract, it's reflecting a specific location. Okay. Now, looking a little bit closer at our spinal cord, we see our gray matter and we see our white matter. And that white matter, remember, is that myelinated axons, and the gray matter tends to be unmyelinated axons, or cell bodies and dendrites. So if we look at our gray matter, we can see that it forms this little H, or this little butterfly shape right here in the center of our spinal cord. And the white matter is outside and surrounding the outside of that gray matter. There's some structures to identify. This is the posterior horn back here. You'll also see it labeled as the dorsal horn, so either one is fine. The lateral horn is on the side here, and then our anterior horn, again, is up front. You can see that the two halves here are connected by that gray commissure. Now, around the gray matter, we see our posterior column, we have our lateral column, and we have our anterior column. And again, our white matter is those myelinated axons, and that's going to transfer information from one part of the central nervous system to another part, while our gray matter is generally where our information processing is happening, so our synaptic integration. Now, while we're also looking at that, we can see our spinal nerves coming off of our spinal cord. And you'll remember our vertebrae are removed from this image, so this is the reason that these spinal nerves are coming out in these designated areas, is because those are passing through the intervertebral foramen. So we can see that we have two branches. We have an anterior root that's leading towards the anterior side of our spinal cord, and we have our posterior root that's leading to the posterior side. And so we're going to get to know those pretty well here. And again, here's that term, ganglion. So a posterior root ganglion, so a collection of cell bodies right in here. Now here is that dorsal root ganglion, that collection of cell bodies, and if you remember, these are going to be from those pseudo-unipolar neurons that are bringing sensory information back in all the way down those axons, and those cell bodies will be here, and that's going to lead back towards that dorsal horn where we had that synaptic integration going on. In front, we see a pair of anterior ventral horns here and here, and that is leading out to our ventral root. So that's going to carry motor information back out to our effectors in our bodies. And then last, we have our lateral horn right over here, and this is where neurons from our sympathetic nervous system are located. Now over here on the right, you can see our afferent sensory information coming in. There's a cell body leading back to this different regions located within that gray matter, those horns. And so we have our somatic sensory nuclei here, we have our visceral sensory nuclei here, we have our autonomic efferent nuclei here. So this can be broken up into different regions based on where that information is coming in. And then up front, more anteriorly, we can see that we have that motor signal coming out of that nuclei. So remember, a nuclei is in the central nervous system, so cell bodies within the central nervous system, leading out to our effectors. Now, if we look at that white matter, we can see that we have those columns we talked about earlier. And in red here, you'll notice that we have our ascending tracts, which are carrying information up, and we have our descending tracts, which is bringing information back down and out. So these axons are kind of bundled together, and they transmit information up and down these tracts. So we have that posterior column, lateral column, and our anterior column here. And so we're going to be looking at different pathways, or those different tracks, if you remember those bundles of axons within each of these columns. And so these columns then can be further divided into little groupings that are coming from different regions or going to different regions. So let's make meaning of what a nerve actually looks like, and let's try to organize this to help you understand this a little bit better. Now, if we look in this bottom right-hand corner here, we can see our endoneurium. Our endoneurium is a layer that is surrounding our axon here in yellow and our myelin sheath around it. And so this endoneurium is going to help prevent that signal from jumping to other nerves. We can see that myelinated nerve. There's a lot of those myelinated nerves right in this image in here, but we also see unmyelinated nerve fibers that are also within this bundle, and we call these bundles fascicles. Now the perineurium surrounds or creates those fascicles and bundles unmyelinated and myelin nerve fibers together. Then we have multiple bundles or multiple fascicles that are surrounded by the epineurium. So we're packaging bundles together, and we call this a nerve. You'll notice lots of blood vessels within those fascicles and in between those fascicles, because we have to provide nutrients and oxygen and remove wastes for all of these neurons. Now remember, information is being transmitted back to the spinal cord in this spinal nerve, but motor neuron information or motor information is being transmitted out to our effectors. So information is going back and forth through these different bundles. As you can see here, there's this split in that spinal nerve. And so we have our anterior root up here, and if you remember, that's where motor information is coming from the spinal cord and going out to effectors. But sensory information is coming back down those axons, past that collection of cell bodies, through this posterior root, through the rootlets, and back into our gray matter of that spinal cord. So let's just go over those terms and put some definitions to them. So our nerve, remember, is an organ. Our nerves are made up of different nerve fibers, and they're bound together by connective tissue. So we have more than one tissue type here. So a nerve is in fact an organ. There's lots of fibers, thousands of fibers going in opposite directions. And in our peripheral nervous system, they are surrounded, those axons are surrounded by Schwann cells, while in the central nervous system, they're surrounded by the oligodendrocytes. Now the endoneurium is that thin sleeve that's going to surround those axons, and so this is loose connective tissue. Those are bundled together in fascicles, and they're surrounded by the perineurium. That's going to wrap around those fascicles. And then our epineurium bundles our fascicles, and we see that this is dense irregular connective tissue. Remember, it's got collagen fibers going in every different direction, and so that's going to help prevent stretching or injury to those nerves. We saw all the blood vessels there that are penetrating and bringing a really rich blood supply to all those neurons. So go ahead and take a few minutes, practice this, use these terms. I'd recommend printing this out a couple times and have multiples laying around so that you can really get to know that terminology well. All right, welcome back. So I hope you were able to label that from memory, but do it again, and do it again. Remember, long-term potentiation here is your goal. So again, this image is just kind of a zoomed in version of what's going on here at this branch. And so we can see here in red our sensory information coming back through that dorsal root ganglion and those dorsal rootlets, and our motor information being carried away. So if we look a little bit closer, we can see our posterior root right here. We can see our anterior root right here. And we can see that epineurium surrounding this. So out here, we call it a spinal nerve. Back here, we're going to refer to it as our posterior root and our anterior root. And we can see all those cell bodies clustered here in that ganglion. Now those spinal nerves are branching out, and we can see that they then have branches coming off of them. So here we can see our anterior ramus, or the branch that is more towards the anterior side of the body. We can see our posterior ramus coming back here, and we can see a meningeal branch right over here, and we can even see branches that are coming over to our sympathetic system. Now if we look at the skin specifically, and we look at the innervation of our skin, we can see that the body is divided into what's called a dermatome, and these are these areas of skin that are receiving that sensory information from those spinal nerves. So in the case of C5, we can see the region that is being innervated by the C5 spinal nerve. In here we can see T1 branching down, and over here T2, T3, T4, right? So these dermatomes overlap their edges, and they can overlap up to 50%. So when you're anesthetizing superficially, you need to actually make sure that you're anesthetizing 3 regions to make sure you get all of the spinal nerves that are innervating those areas. So take another minute, just practice again real quick, label this diagram, see how well you did. All right, now what we want to do is we want to build some connections. We want to take what we've gone over and we want to really see the bigger picture, and we want to step back. It's kind of like looking at Google Maps. You know, you look up an address, it shows you, it kind of zooms into that location. When you hit directions, you zoom out, and we can see the pathway now on how to get to that location. And that's kind of what we're doing with the next half of this lecture. So if you remember ascending tracts, or ascending tracts are those bundles of axons are going to be carrying sensory signals up our spinal cord. And that is going to pass through 3 neurons from the origin of those receptors back to different parts of our brain. So our first order neuron is how we'll refer to them, pick up the stimulus. And so this is what, when we think about, oh, we touch something, it hurts, that signal is transmitted down back into our spinal cord, and that's going to be our first order neuron. That's that pseudo-unipolar neuron that we've been talking about, or the afferent neuron that's transmitting that sensory information. Our second order neuron then passes up to the thalamus, and then eventually takes the signal from the thalamus, which is our relay area of our brain, to some part of our cerebral cortex. And so that is going to be transmitted by our third order neuron. It's taking that signal from the thalamus to the appropriate region in the cerebral cortex. So here's that image again that we looked at. You can see in red, we have our ascending tracts. This is in that posterior, lateral, and anterior column. So those columns are made up of white matter, so myelinated axons. So our first pathway that we're going to look at is this Gracile Fasciculus. So this is going to be transmitting information from receptors that are going to tell us about body movement, our limb position, fine touch, and pressure. So this is going to carry signals from our mid-thoracic and lower parts of our body. We're going to take information relating to vibration or pain in our organs, fine touch, deep touch, proprioception from our limbs and our lower trunk. And so proprioception is basically our sense of position and movement in the body. Now our first order neuron is going to take that information from those sensory receptors back to our spinal cord, all the way up into our gracile nucleus in the medulla oblongata. Where are we? Here's our medulla right over here. You don't need to know the gracile nucleus, but if you remember, a nucleus is a collection of cell bodies, but that is in our central nervous system. So basically, when you are sensing information in your lower limbs, it's passing up a single neuron from your lower limbs into your spinal cord all the way up to your medulla oblongata in your brainstem there. From there, our second order neuron will pass from that region over in our medulla oblongata up to our thalamus in our brain, our relay center, and from there we can see that it synapses with our third order neuron that transmits that signal to our post-central gyrus, or our somatosensory region in our brains. So that information has to get all the way up to here from our lower limbs or organs, and so it's going to pass through just those three neurons along the way. So as you're practicing this, think first order neuron from our sensory receptors to our medulla oblongata, our second order neuron from our medulla oblongata to our thalamus, and third order from our thalamus to our post-central gyrus. The spinothalamic pathway then are receptors for pain, heat, and cold. So this is coming from all segments of our body. And so we're bringing in signals from our skin, for pain, for pressure, temperature, light touch, tickle, itch, right? So when we talked about all these different receptors with our integumentary system, we talked about our mechanoreceptors that are responding to pressure and touch, so our Pacinian, our Merkel, and our Meissner, the corpuscles. When we talked about thermoregulation and picking up temperature, that's going to come in through this spinothalamic pathway. Nociceptors, noci means hurt, and so basically these are our pain receptors. So our first order neuron leads from wherever these receptors are being stimulated, and they're transmitted back to our posterior horn, and you can see first order neuron leading back to that posterior horn where it synapses in the posterior horn of whatever that branch or that spinal nerve is. It's transmitted up the spinal cord back to our thalamus, our relay center, and from there it synapses with our third order neuron back to our post-central gyrus in that cerebral cortex for us to process and figure out some kind of meaning to it. So let's quickly review and trace these pathways back to the control center. And what I want you to think about is naming every structure along the way and where are we located as you go through that process. And what division are you in? Are you in the peripheral nervous system or are you in the central nervous system? Okay, so let's take a look at this, and I'm just going to jump ahead a slide, and then we're going to come back to that slide that I just showed you. Now, remember, we have all these different receptors. So we've got these nerve endings, free nerve endings. We've got information coming from our hair follicles. We've got our corpuscles here. You can see our Pacinian corpuscle, our Merkel cells, our Meissner's corpuscles. This is going to bring in information. It's going to be sent back into different parts of that gray matter, different parts of that dorsal horn, which then comes up to our thalamus and back to our primary somatosensory cortex. Okay, so what is happening here with that signal? All right, we are stimulated, and someone taps on our skin, and that stimulates our mechanoreceptors. Those mechanoreceptors create an excitatory, an EPSP, in our neurons, and that is going to then reach threshold, which then initiates an action potential, our depolarization, repolarization, hyperpolarization, and return to resting membrane potential. That action potential jumps down those nodes of Ranvier, and we call that saltatory conduction. That signal is sent down our afferent sensory spinal nerve. This is our first order neuron of our spinothalamic pathway that passes in through our intervertebral foramen, that goes through our dorsal root, past our dorsal ganglion, through our dorsal rootlets, and into the posterior horn of our spinal cord. And so there we have a synaptic cleft where we have the release of neurotransmitters that are going to, if a large enough signal, are going to stimulate an EPSP. That EPSP is going to then fire another action potential in our second order neuron of our central nervous system. It's going to lead up our spinal cord back to our thalamus. Here in the thalamus of the brain, our brain is processing that information, and it's our relay center. It's going to send it to the right spot. We have a synaptic cleft, again, where neurotransmitters are released. We stimulate an EPSP and then an action potential in our third order neuron that takes that information up to our postcentral gyrus in our central nervous system. Whew, that's a lot, right? But if you go over this again, you're tying in the information we talked about in Unit 3 with a pathway that we're learning in Unit 4. Now, when it's in our brains, we can see that information is connected within our brain as well. So we have these projection tracts that are connecting different parts of that cerebral cortex, or maybe from a higher to our lower brain. We can see that we have these commissural tracts that are connecting the two hemispheres, mostly through that corpus callosum. And we have association tracts that are connecting different parts of the same hemisphere together. So now what we're doing is we're sending that information that's coming in to different regions in our brain to help make meaning of what is going on with that information. And then our brains can send out information through our descending tracts. And so this is going to carry motor signals down through the brainstem and the spinal cord. But this only involves two neurons. So our upper motor neuron, where it's going to start in our cerebral cortex or maybe in our brainstem, and ends on a lower motor neuron. And that lower motor neuron is going to come out of either the brainstem or the spinal cord and pass out to our effector. And that is going to lead to our muscles or our organs. So again, here's those green descending tracts, again in those columns, because we're going to be transmitting information along those descending tracts. And so the first one that we'll look at is called the Corticospinal Tract. And so most of our neurons from our precentral gyrus in that cerebral cortex are going to be responsible for our voluntary motor control, right? So when we're trying to move. So there's our two neurons in our pathway. So that upper motor neuron coming from the cerebral cortex all the way down to our lower motor neuron that's going to branch out. And depending on which muscles we're trying to stimulate, depends on which branch or which spinal nerve, that information is going to be transmitted out. If we're trying to move our arms, that signal is going to come out up here more superiorly, but if we're trying to stimulate our legs or move our legs, that information has to go much further. So take what we just did with tapping on our skin, and let's say someone taps on your arm or your skin and you want to move your arm. So how does that information go back? All right, so how was it? Did you do all right with that? Now, just to show you an image, here is that upper motor neuron coming out of that cerebral cortex, passing down through our spinal cord, synapsing with our lower motor neuron that's passing out to our muscles. So here's that first slide again, showing you that pathway of sensory information coming in from the mechanoreceptors, leading through that first order neuron, back to our second order neuron in our thalamus, and then up to our post-central gyrus from that third order neuron. Now, once that information is processed and we decide we want to move our arm, then that precentral gyrus is going to send a signal back through our upper motor neuron in that central nervous system of our corticospinal tract. That then synapses with a lower motor neuron, and I would encourage you to practice how an EPSP and an action potential is transmitted every time you talk about this. That lower motor neuron then is going to carry out that motor information through that spinal nerve. So that's going to be part of our peripheral nervous system. So it's going through that anterior rootlet to our anterior root, out through that intervertebral foramen into our spinal nerve, and then that's going to innervate to our flexors and our extensors, and we move our arm away from that tapping. So in Unit 5, we're going to really focus on that connection between our spinal nerve and our flexors and extensors, and we'll call that our neuromuscular junction, and we'll learn how these are stimulated. So let's look at a few more of these descending tracts. First one we'll look at is our Tectospinal Tract, and this is coming out of our superior colliculus. Together, there's two of these. They are referred to as the superior colliculi. You'll learn about these in the lab when you do your brain dissections. They're responsible for receiving sensory information from our eyes and sending out movement. So it helps us orient our eye movements to objects that we're interested in. And it's also involved in our reflex when we're turning our head in response to some kind of sight or sound. And so you can see down here that this information is exiting C1 through C4, our spinal nerves C1 through C4. So this is going to be responsible for moving some of those neck muscles that you learned about in lab. Another one that you'll learn about, or that you should know about, is the reticulospinal tract. This is going to help control muscles in our upper and lower limbs and is responsible for posture and balance. And then our vestibulospinal tract is bringing in information from our inner ear, and so that's helping us again control our muscles and our limbs for balance. So we can look at this over here. We can see we've got these muscle and joint proprioreceptors. We're getting information in about our position in the world. We have information coming in from our inner ear. We have information coming into from our eye, and that information is leading into our cerebellum. We also have information coming in from our motor cortex. All that information is being processed by the cerebellum, and the cerebellum is then responding to our muscles through the reticulospinal and vestibulospinal tracts to help keep us balanced and help us with our posture. But it's also sending signal back to the brain so that the brain is aware of our position as well. Now, maybe some of you took the ice bucket challenge a handful of years ago. in order to help raise money for ALS. Now, what ALS is a destruction of these motor neurons, and that as a result, our skeletal muscle can atrophy or shrink because there's a lack of innervation and a lack of stimulation, and ultimately a lack of use. So ALS, also called Lou Gehrig's disease, is a sclerosis or a scarring. If you remember, we talked about multiple sclerosis where the myelin sheath gets damaged. Here we can see that it's again a scarring on the spinal cord. And so if we're scarring that spinal cord, that scar tissue doesn't function appropriately like the regular normal tissue does. And so here we see that astrocytes are failing to reabsorb glutamate, which is excitatory if you remember, and that can have extra stimulation on those postsynaptic neurons. Some early signs are muscular weakness, difficulty speaking, swallowing, or the use of hands. But as the body and these motor neurons tend to break down, we lose our ability for normal functioning, but it rarely affects our sensory and our intellectual function. So sensory information can still come in, brain can still work fine, and Stephen Hawking is an excellent testament to that ability to keep his intellectual function going. So that is the end of today's lecture. I hope that you were able to kind of see where we're going and why we're connecting all this together, because it really does connect this entire class together through these pathways. So before next lecture, spend some time reviewing those pathways, getting used to the structures of spinal nerves and the organization of spinal nerves. And then what we're going to do in our next lecture is tie in some reflexes and our special senses. So our sight, our taste, our hearing, and see how that also transmits throughout our body. Have a great afternoon. See you next lecture.